The last speaker of our session is Sakshi Sambhavi, who will discuss on mitochondria, an isolated yet socializing organelle, and how this socialization aids in maintaining the overall cellular homeostasis.
Good morning all. Good morning all. Starting with a quote. Sorry? Speak little louder. Okay. So, life on earth is more like a verb. It repairs, maintains, recreates, and outdoes itself. So, people in the audience who are thinking, when did Sakshi turn into a philosopher? It's not me. These are the famous lines by Dr. Lynn Margulis, who was a great scientist who actually discovered or proposed the endosymbiotic theory, which marks actually one of the first events in the history of eukaryotic evolution. She proposed that the bacteria, a simple proteobacteria got engulfed by an archaean, which stayed inside, developed this endosymbiotic relationship with the archaea, and eventually dividing and evolved into the current day mitochondria. So there are various theories or models which are proposed to explain this theory, and I'll be talking about one of the most accepted and the recent theory that has come up. So this is an inside-out model which explains the endosymbiosis event. It starts with a simple archaea which was living in a close association with a bacteria, and slowly the archaea started developing protrusions and covering the bacteria, so these protrusions, when they were covering or enwrapping the bacteria, they actually started exchanging materials as well, and ultimately leading to expansion of these blebs and formation of the nucleus, the endomembrane system by these expansions, ultimately fusion of all these, the blebs surrounding it, and leading to formation of the plasma membrane. And here we have the first eukaryotic cell, which is having the mitochondria itself, which came from the bacteria, and the plasma membrane, nucleus, and all the endomembrane systems evolved. So now the bacteria, you can imagine a bacteria which is coming from outside, and it is just trapped inside a cell or inside a host. Now it has to survive, right? So it has to develop some features or things or modes of communication in order to socialize with its neighbors, which are in a way the organelles inside as well as the host. So today I'll be talking about the rise of mitochondrial sociology, where how and what all means the mitochondria or the bacteria, the ancestor, has adapted in order to communicate or socialize with all the organelles inside the cell. So the possible mode of communications, and since we are talking about something as old as almost 2 billion years ago, of course pigeons were not there for mitochondria to use. So the only options left was either it could use the distal mode of trafficking, where the cargoes are packed inside the vesicles and they might be going and fusing with the mitochondria, or a proximal mode where the mitochondria itself goes and makes contacts with the other organelles inside the cell. So I'm giving you a very typical picture of a vesicular trafficking system. So this is a picture which we have been seeing from our school days, right? And I would like to ask you a question. Have you ever wondered where is mitochondria in this picture? And the answer is no. Mitochondria is not a part of this endomembrane trafficking system. And this was one of the biggest questions, as in, we know that mitochondria has to rely on other organelles for its demands of lipids, proteins, ions, and everything. So how come mitochondria is surviving? How is it exchanging the materials from the outside world? So it looks like mitochondria had some other plans. So in 1956, there was this observation made where mitochondria and endoplasmic reticulum were seen to be existing in a very close proximity. But this striking observation was actually ignored for almost 50 years. And only recently, in the last decade, when people are really going inside the cell and looking for all the organelle dynamics, they realize that it's not only mitochondria and ER, but also almost all the organelles inside the cell have this dynamicity where they come in close contact with each other, and also even these are not just mere physical contact, but a functional contact as well. So this was a very new emerging field, and even people in the field didn't know how to define it. What is a contact? So all the stalwarts from the cell biology field, they came together to define membrane contact. It was recently in 2019. So they have come up with certain characteristics of what to call a contact or a membrane contact site. One being tethering, as in the two organelles coming together have to have a tethering mechanism, as in a protein might be involved in holding both the organelles together. The other is lack of fusion, where the two organelles which are coming close, though they are closer, but they are not supposed to fuse. So the only contact that we call is the one which is not having a fusion, and there has to be a particular distance maintained between the two organelles. And in the field, the most widely accepted distance, if I can give a number, is almost 30 nanometers between the two organelles. That has to be there for it to call a contact site. And of course, the two organelles cannot be just coming together and for chit-chatting. It has to have a specific defined function in order to call, and we'll be seeing what all functions a mitochondrial contact site can have. And the other one, people recently are talking about a specific proteome as well as a lipidome, which has to be a characteristic of a particular contact site. So these are a few of the characteristics and of a membrane contact site. Now moving towards what all mitochondria could socialize with, and how these contact sites are helping it function better. So one of the major thing that the mitochondria uses contact site for is the lipid transport, where we know that it has to rely for the demands of lipids with the other organelles. So there are lipid transporters which actually transport lipids from endoplasmic reticulum to the mitochondria. We'll be seeing an example of it further. And again, calcium signaling, as we all know that calcium is there in the is produced in the ER, and any other organelle, let's say mitochondria as well, whenever it needs calcium, it has to take it from the ER. So this marks the ER mitochondria contact site. Also, ER and mitochondria contact site has been reported to be very important for the dynamicity of mitochondria, where even if the mitochondria has to divide, the fission process is also marked by the enwrapping of endoplasmic reticulum around mitochondria. This was something new. And the other interesting aspect that we have is not only two contacts coming together, but it's a three-way contact where endoplasmic reticulum, plasma membrane, and mitochondria come together, form a contact that helps mitochondria to be fetched from the mother cell to a new daughter cell, thus helping in the mitochondrial inheritance as well. So these are some of the functions of a mitochondrial contact sites. And now so keeping in account the architecture and the function of a contact site, people have been able to identify what all ways are there for mitochondria to socialize, and looks like mitochondria contacts with almost all the organelles inside the cell, and these are several names of the different contact sites that has been recognized. And I want you to note that since this is a very new field, so though all the contact sites have been identified, but it is not fully characterized in terms of its proteome, in terms of its lipidome, and not even in terms of its actual function that these contact sites are doing. Of course, there are one or two contact sites, for example, ER mitochondria contact site and mitochondria vacuole contact site, which are characterized pretty well, but still there are much more to come. Now I will give you one example of where the field is heading. So this is an example not from a mitochondrial contact site, but ER lysosome contact site, where there is a protein lipid transporter which transports lipid from endoplasmic reticulum to lysosome. So people are trying to actually capture the contact sites at work using cryo-electron tomography and correlative light electron microscopy, where people have tried to get the density for this protein called this lipid transporter VPS 13, and they have modeled even the AlphaFold predicted model in the density, which actually shows it is fitting well and gives a beautiful representation of how a lipid molecule can be transported from one organelle to the other organelle using this hydrophobic cavity which is there inside. Sorry. So this was all about the architecture, function, and where the field of the membrane contact site is heading. Now I'll ask a question from an evolutionary perspective. When did contact site emerge in the course of eukaryotic evolution? So the answer for this question lies in the inside-out model itself, which I have already explained, where when we were talking about archaea and bacteria forming a close association with each other and exchanging lipids and metabolites, it was nothing but contact site or the membrane contact site, which eventually led to the transfer of materials and exchange of materials from archaea and bacteria, eventually leading to evolution of the eukaryotic cell. So now, since we are talking again about the evolution, one thing that was so striking and puzzling to think that bacteria and in fact all the other three domains of life are known to shed vesicles, right? Even bacteria forms extracellular vesicles in order to communicate with the other bacteria. So how can someone imagine that a bacteria which was engulfed by an archaea in the course of evolution when it evolved into a mitochondria just lost its capacity to form vesicles, and that too at a time when it required it the most, because it could have used these vesicle shedding capacity in order to communicate with the host. So these are few of the examples where all the three domains of life have been known to shed vesicles. So now, turns out that that is not true. Mitochondria, again, they do shed vesicles as that of their ancestor bacteria. So this was recently recognized, identified by Dr. McBride in 2008, where she saw that mitochondria also can shed some of the vesicle-like things, and they now have compared even the bacterial vesicles with the mitochondrial derived vesicles, and looks like they all have the same kind of vesicle formation as that of bacteria. So though this is very new thing, but people have recently figured out, or actually trying to figure out, where all the mitochondrial derived vesicles are used, and looks like the mitochondria shed some of the vesicles for it, so that it can be fused to the lysosome in order to get rid of the oxidized proteins or the lipids. The other process where it is employed is the mitochondria sheds vesicles which fuses with the phagosome and helps killing of the microbe or the bacteria inside the phagosome. The other interesting thing that MDVs are employed in is in mitochondria to mitochondria communication. So as that of bacteria, it secretes these vesicles which goes and fuse with the other damaged mitochondria and helps it repair. So one thing that I would like you to note is that whatever vesicles we have seen in terms of the mitochondrial biology is only coming from mitochondria. There is no such report where mitochondria can accept a vesicle. So this is only a one-way route from mitochondria to some of the other organelles inside. And so the fact that mitochondria is not a part of vesicular trafficking still holds true, because it cannot accept vesicles from outside. So this is the current picture of mitochondrial sociology that we know of. The most important mode of communication that mitochondria employs is membrane contact sites, where it goes and communicates with the other organelles by forming a contact, physical contact as well as a functional contact, in order to exchange the lipids and other materials. The other thing which it has derived from its bacterial ancestor is that shedding of vesicles and using it to contact some or the other organelles inside the cell, specifically the mitochondria itself and the lysosome. So since all these things are very new in the field, so there are a lot of questions still unresolved. One being, which is not yet explored and not answered in the field, is why is mitochondria not a part of vesicular trafficking? And given the dynamicity of the vesicles, how is mitochondria kept isolated? And I believe that there are still unexplored ways how mitochondria can socialize. So I'll just leave you all with these questions. Thank you.
Thank you, Sakshi. Now we can take some questions.
Hi, Sakshi. I have a question. Regarding, since we are talking about contact sites, are two mitochondria or different mitochondria connected in cells? And the second question is, how dynamic are mitochondria in a cell?
How dynamic are, okay, I didn't get your first question. Okay,
Are mitochondria connected in a cell? Two mitochondria or different mitochondria within a cell, are they connected?
Okay, so regarding our first question, how are mitochondria connected inside the cells? So one thing is mitochondria actually are very dynamic in nature, where they fuse. The fission and fusion process is very dynamic and continuously happening. So in fact, if you see, mitochondria is more like a thread, where the individual mitochondria again fuse with each other to form this thread-like structure. That is one. And coming to your second question, how dynamic? So this is, I guess, this is the answer for that too.
Yeah, I wanted to ask, what is the status of these mitochondrial contact sites in dormant cells?
Dormant cells.
Yeah, when the cells undergo dormancy, do they increase or they reduce or what?
Okay, so I am not sure about the dormant cells, but I haven't come across any such thing. But I can tell you about the stress conditions, because dormancy is again a stress. So there people have seen in different stress conditions, for example, in glycerol or even other stress conditions, mitochondria form more networks, or the dynamicity increases even more there.
Since we know that mitochondria are double membrane organelles, right? So addressing the first question, my thoughts are maybe as in your second slide you showed that mitochondria escaped the endovesicular system.
Yes.
It evolved simultaneously along with the endoplasmic reticulum.
Yes.
So would it be correct that since it escaped from the endomembrane system, that may be the reason the mitochondria is not a part of a vesicular system, and it is not forming a vesicular network while forming its social networks with the other organelles? Can it be the reason? What are your thoughts?
Okay, so what are my thoughts? So when I talked about mitochondria escaping this system, so it is again as that of bacteria, because even bacteria are known to escape these endomembrane systems, and I don't think that by escaping, I just mean that it just escaped through that membrane, right? So that is, I don't think that is a criteria for it to not be part of a vesicular trafficking. But what I think, why is it kept isolated from vesicular trafficking? This is just my thought. I don't know what is the actual thing. Maybe because mitochondria has to maintain a membrane potential, right, for it to work. Even the ATP synthase and all the complex processes happening there. So whether the fusion of the vesicles will interfere with the membrane potential, that is very much required for normal mitochondrial function. So that is what I think this might be one reason why mitochondria is kept out of the system, the endomembrane system. Yeah.
Thank you.
Sakshi, you are aware that there are endosymbiotic bacteria in, say, like Wolbachia and so on with insects. So do they use some of the same kinds of contacts that have been identified for mitochondria, and whether it's a repurposing of the same tools that were previously useful, are they being reused for these endosymbionts? Has anyone looked for this kind of a possibility?
Yeah, so I tried to look for where any such evidences are there, but I could not find any such thing. But what I have come across is there are some transporter proteins. For example, the one I showed, VPS13, which is employed at the contact site in order to transport lipids. So there are some remote homology seen in case of bacteria, where they also have protein. Some of the proteins which have homology with them.
The archaeal-derived proteins, because when the bacterium, these are much later
Yes.
symbiosis than the mitochondrial symbiosis,
Yes,
right?
yes.
But they do have a fairly intimate thing, and they go down the female germline,
Yes,
right?
yes.
So whether the archaeal-derived proteins that serve for your contact, like that very nice
yeah.
picture you showed, whether they are again utilized to trap these endosymbionts.
Okay.
Since now some
Yeah.
proteins have been found, one can look in these cells to see whether those are expressed and they are in the site of where the endosymbiont is also sitting.
Yeah,
That's-
sure, yeah. I tried to look for, but I could not find one. Okay, thank you.
The speakers will be available till the evening, so if you want to discuss with them, you can discuss directly. So now we are breaking for high tea, which is arranged in CCMB Shantiniketan. Please assemble back here by 11:25 for the panel discussion. Thank you.